Think about all 46 chromosomes in the human body. If the DNA from all were laid out end to end, it would measure roughly 2 meters. DNA must be tightly packed in order to fit into the cell's nucleus. However, it must also be readily accessible for gene expression. The highly compacted chromosomes that are viewed during metaphase are called metaphase chromosomes. In this animation, we will examine the compaction level of metaphase chromosomes.
In the first level of compaction, there are repeating structural units located within the eukaryotic chromatin. It begins with the short stretches of the DNA helix that wraps itself around the histone octamer. The histone octamer is an 8-protein complex that is located at the center of a nucleosome core particle. The histone proteins attach to the DNA helix with the use of electrostatic and hydrogen bonds. Within the histone octamer, there are 2 copies of the 4 different histone proteins, making it a total of 8 histone proteins, and 1 linker histone.
There are 5 types of histones. 4 core histones that include H2A, H2B, H3, and H4. 2 of each of these histones make up the octamer, and attached is a flexible charge amino terminus, also called and looks like a tail. The last histone is the linker histone, known as H1. The H1 binds to DNA, can help compact adjacent nucleosomes, and it is also less tightly bound to the DNA than the 4 core histones. When all of the histone proteins come together, it forms a bead-shaped histone DNA complex, referred to as the nucleosome.
The next step of compaction relies on the chromatin or chromatin fibers. In this step, the packaging of DNA into nucleosomes shortens the fiber length so that a piece of DNA that is 1 meter long will become a string of beads chromatin fiber that is just 14 centimeters long. Therefore, chromatin is further coiled to an even shorter, thicker fiber called 30 nanometer fiber. Basically, this shortens the chromosome so that it's about 50 times shorter than the extended form, and this is usually displayed by a spiral or zigzag model. Also, the stability of the structure requires the presence of histone H1.
The 3 nanometer fiber is then anchored to the protein filaments, forming the radial loop domain, and then attaches to the internal nuclear matrix. The nuclear matrix is a fine network of irregular protein fibers and other proteins bound to the protein fibers. The nuclear matrix is made of two separate parts. One is the nuclear lamina, which is made of fibers lining the nuclear membrane. The other part is the internal nuclear matrix, which fills the interior of the nucleus that has the proteins bound to it. In the formation of the radial loop domain, the 30 nanometer fiber will form a loop as it attaches to the protein fiber from the nuclear matrix. The 30 nanometer fiber and the nuclear matrix bind with proteins on the internal nuclear matrix. The area of binding is called the matrix attachment region.
Now that you have a better understanding of the steps in eukaryotic chromosomal compaction leading to the metaphase chromosome, let's see if you can answer this question. Describe what structural changes convert a region that is 300 nanometers in diameter to one that is 700 nanometers in diameter. The answer? In the conversion of a 300 nanometer fiber to a 700 nanometer fiber, the radial loop domains become more tightly compact, which would mean it increases the diameter.